Hi everybody, my name is Christopher Collison, and in this short video we'll talk about a review of the upcoming experiment, the determination of a concentration of food dye in a sports drink. We'll go through a number of slides and we'll also talk about some of the glassware that you can use for the dilutions that will be so important for this experiment. So thanks for listening. So here's the title of the lab that you're working on. Perhaps a better way of describing it is simply to say how much of this red stuff or blue stuff do we have in these sports drinks bottles. Before we get started, I'm going to just remind you of a few things that were absolutely critical as we're moving through any experiment. So first of all, you must keep a lab notebook. The key idea here is write everything down. Your ideas are very valuable, your observations are very valuable, and paper is effectively very cheap. So write your ideas down. They're going to be very helpful for you as you write up your lab report. Second thing I want to remind you of is that you will need to submit an abstract for the work that you're doing and for the report that you write. And that abstract is going to be what gives you at least half of your grade for the experiment. Keep your abstract clear and concise, and also make sure that you proofread it before you hand it in. Before you even get started on your experiment, you have to ask the question, what's your objective? And that should also be written in the top part of your lab notebook. In this particular case, we know that the objective is to calculate how much of these particular dyes are in the sports drink that you care about. The objective is the why. Why are you doing what you're doing? It's the thing that attracts the reader to keep looking at your report. It's the thing that motivates you to actually go through and complete your experiment. And when you have a procedure that's tied to your objective, you know that you're on the right track. So in this case, specifically, what are we trying to do? We're trying to calculate the number of moles of a particular dye in a particular sports drink, again specifically in a 20-ounce bottle of the sports drink, or in fact, the serving size that's written on the back of the sports drink. You'll also need to calculate the number of milligrams of that particular dye in that particular serving size and in that particular 20-ounce bottle. The next thing for you to think about is how will you do your experiment? In this particular case, we want to determine, first of all, the molar absorptivity of these standard dyes. What we're going to do is measure the absorption of the standard stock solutions that you get. And from that, if you know the concentration, if you know the mass of the dye in that particular volume of solvent, then you can calculate something called the molar absorptivity. And when you have the molar absorptivity, then that will tell you in an independent measurement, it will tell you when you measure the absorption, for example, you divide that by the molar absorptivity, and that's going to give you the concentration that you're looking for. So what is your procedure going to look like? You're going to be given some standard stock solutions of these known sports dyes. So we can get these molecules, we can buy these molecules online. We know they're in pure form, and we can build a solution, set of solutions that we put into the UV-Vis instrument, and from there we calculate the molar absorptivity. That's been described to you in a prior video, so I'm not going to go through it again here. But ultimately what you're trying to do is determine that molar absorptivity. How will you do that? You need to dilute your stock solution. So this experiment is largely about dilutions, 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 and more dilutions. And I think everyone is short of time. In particular, we'll be short of time in this lab course. So we have to make every second count. How can we do that? So the first thing we might do is we might dilute in a very quick and dirty way a small amount of the standard, kind of get an idea that if we dilute by a factor of 100, what kind of absorbance are we getting? What kind of absorbance can we measure in the UV-Vis instrument? So very often a quick and dirty experiment can save you a lot of time later. Sometimes we actually start putting forward a grand procedure, and that gives us a very slow experiment that ultimately may not work out to be quite so accurate. So we may often find a slow and ineffective procedure can be prevented by simply running a quick experiment. And I encourage you to do this. Talk to your instructor. Think for yourself, how might I take a small quantity, dilute by a known large quantity, and kind of see what kind of absorption we get. We definitely want to be below 1.5 in the instrument. What are the materials that we're working with? Well, blue number 1 is also known as brilliant blue, and you can see the molecular structure of this particular material on the left hand side, along with the molecular formula. Of course, from that molecular formula you can calculate the molecular weight and therefore the molar mass. We also have a material called red number 40. This is also known as Allura red, and that's shown in the panel to the right hand side, with again the molecular formula being available to you below. Now, how do we do these dilutions? And what I want to do now in the next few slides is introduce you to a few of the items of glassware that you might find around the lab. The volumetric flask is a very good piece of glassware to use when you're doing your dilutions. It's particularly useful when you need a large volume of these solutions to carry on your experiments. In this case, in this particular experiment, you may only need 3 or 4 milliliters of a particular solution, and so perhaps the volumetric flask may not be your best approach. So let's just go through some details of when the volumetric flask is best for your experiment. It's useful when a large volume of analyte will be required and comparable to the volume of the flask. That means if the volume of the flask is 250 milliliters, then this is ideal for an experiment where you'll need something like 200 milliliters to run the experiment. So if you only need 3 or 4 milliliters, perhaps this approach is not the best. The volumetric flask is also useful when the solvents are abundant. So water is relatively abundant, and so it might be a good choice when we're working with water-based solutions. However, we also have to think about how we waste liquids, and if we are creating a large amount, 250 milliliters or a liter of material, that material has to be wasted and has to be manifested somewhere, and there's a cost associated with hazardous waste. So we have to keep that in mind as we choose our glassware. So definitely do not use the volumetric flask if you're going to do successive dilutions. This basically means you keep adding more and more volume, and that volume is unnecessary in most cases. So don't use a volumetric flask when solvents are costly. You might find at some point in your career that you use something like chloroform. Chloroform is an expensive solvent. It's also a hazardous solvent, and so we don't want to use more of it than we really need. Next, we're going to talk about the graduated pipette. And there are lots of graduated pipettes that are around the lab. On one particular end in these green buckets, you'll find some graduated pipettes. And unfortunately, these days we tend to use these pipettes that are considered to be disposable. You can certainly wash them and use them and reuse them if you're using the same solution. But of course, we also don't want to contaminate the solution if we leave water droplets in a graduated pipette. So be thoughtful of how you want to use these, and certainly for more accuracy you want to use a new dry pipette each time. So these are useful when we're measuring aliquots of stock solutions. Often we take stock solutions in small volumes and we add them to large solutions or large volumes of solvent to reduce the concentration and make our solutions more dilute. That will certainly be necessary in this experiment. Graduated pipettes can also be useful when we're taking small volumes of a stock and adding them to small volumes of a solvent. You can use successive aliquots, one of the solvent, one of the stock solution, if you're just trying to tweak the concentration of your solution. These are also useful when solvents are scarce. So in particular, if we're using chloroform won't dissolve the glass. We're only using a small amount at a time. So be thoughtful again as to what glassware you're going to use. Certainly, you don't want to use successive aliquots when you're measuring solvent into a solute when you need to perhaps add 250 milliliters. If you can only pipette 1 milliliter at a time, then 250 times that is going to take you a long time, and also it's going to contribute a lot of error. Next up, we have the pipetteman or the pipetter, and this is very useful perhaps when we want to go from when we want to accurately go from a very concentrated stock solution and we only have a small amount of solvent to work with. So we might be able to take several microliters of a stock solution and add a few milliliters of solvent to that stock solution in order to get a large dilution factor. So generally these won't be used in this lab, but they are very useful when solvents are scarce. And again, we don't want to use these pipetters when we're working with large volumes. One other piece of glassware you might find useful are scintillation vials, and these are not quantitative pieces of glassware, but these are receptacles that we can place aliquots of our solutions, our stock solutions, or our solvents into. So they're valuable perhaps when solvents are likely to evaporate. We can put the cap back on. Again, we just need to remember, if we're working with small volumes, these are fantastic, but they're also non-quantitative. Sometimes we might find they have residual contaminants in that come directly from the factory that certainly have some pH value, or they might be somewhat basic. And also these scintillation vials, even though they're consumables, we must recognize that they cost money to buy, and also if we're using plastic lids, we want to reduce the amount of plastic that we use as much as possible. So be thoughtful about what you use. These certainly work very well in some experiments, but of course we also must consider our carbon footprint, and that's an important factor when we're considering chemistry in the 21st century. So there we have it. Think critically, question everything, and try to visualize what the experiment looks like before you get into the lab so that it runs about as smoothly as you can expect. This will save you time in the end and will give you much better results overall. Good luck, and thanks for listening.